Okay, in this lab, we're going to be focused on the anatomy of the digestive system. Now, I want you to be aware of something. There will be one lab that will focus on the anatomy and one that will focus on some of the physiological aspects. That'll come later on. Right now, what we need to know is basically the structure and how that relates to the function of the digestive system. So let's move ahead. Our objectives here are going to review the gross anatomy of the digestive system. And in each case, this may include some slides and models. Most of those will be coming from what's in the PowerPoint here. We will also be able to look upon the lab exercise text, the Syracuse text, as well as, if you have time, which I really think you all should plan on this, and that is to go over some of the videos that are in the Blackboard for this lab exercise, because that will help review and connect further your understanding of the anatomy and microanatomy of the digestive system. Now, I want to keep this clear also. We're not just going to focus on the digestive system, but it's also its accessory digestive organs. Then we will have worksheets, and those will be located in the Blackboard, and they are due by Friday of this week, 5 p.m. Now, when we start off, we're going to be examining the organization of the peritoneum. The peritoneum, otherwise known as the peritoneal membrane, is the serous membrane associated with the abdominal pelvic cavity. Now, of course, there's two parts. One is the parietal, one's the visceral. The parietal covers the walls of the abdominal pelvic cavity and is continuous with the visceral. The visceral, though, covers the outside of most of the abdominal organs. Keep in mind that many of the organs, in essence, hang or freely float around inside of the abdominal cavity. Some are partially attached into the body wall, the posterior part of the abdominal wall. These are called retroperitoneal, but many are actually attached to the mesenteries, the double layers of the parietal peritoneum. Let's go over some of the different types of mesenteries. The dorsal mesenteries include the greater omentum, the mesocolons, and the mesentery proper. The greater omentum is attached to the greater curvature of the stomach and inferiorly then forms a covering for the small intestine and abdominal viscera. It also loops around, travels superiorly, then posteriorly, and attaches to the transverse colon of the large intestine. The mesocolons suspend two sections of the large intestine, the transverse colon and the sigmoid colon, to the posterior body wall. The mesentery proper suspends the second and third portions of the small intestine, that would be the jejunum and the ileum, to the posterior body wall. Now, the ventral mesenteries consist of basically the lesser omentum and the falciform ligament. The lesser omentum is attached to the lesser curvature of the stomach, while the falciform ligament attaches to the liver and connects the liver to the diaphragm. As I mentioned before, there are some retroperitoneal organs. They're behind the peritoneum. These organs are positioned along the posterior wall. These include the pancreas, the duodenum, and the rectum. Here is a model that you can consider, and you can see the liver, the stomach, the transverse colon, the descending colon, part of the ascending colon, and the entry point, which is called the cecum. You have, of course, the ileum, the jejunum, and, of course, kind of hidden in part is the duodenum, which is attached to the stomach. Now, if you look at these organs from less of a model than from the actual mesenteries, you'll see where these mesenteries exist, and actually, because they kind of flop in front of others and get in the way, some of them have to be trimmed a bit here. This is the lesser omentum. This is the the cut edge of the greater omentum. Notice that they're attached to the stomach. Here would be the duodenum. And then from that, you would have, of course, the small intestine, first the jejunum, and then later the ileum. They are all attached in part to the mesocolon. We also have the transverse mesocolon here, which is attached to the transverse colon. These are all parts of the large intestine, by the way. So we have the ascending colon, transverse, descending, the sigmoid, and eventually, of course, the rectum. And you can see how some of these membranes are stretched out and play a role in support while the organs can kind of move around and jostle around in the midst of the digestive process. If you look from a sagittal section, you can see some of the importance here. Here's the liver, and we have the falciform ligament, which then directly attaches it to the diaphragm. We have the stomach, and of course you have another ligament here. We have basically some of the greater structures here. Pancreas, duodenum are examples, as I said before, of retroperitoneal. We have the parietal peritoneum here. And if you look here, you have small intestine, and it's attached to these various membranes here. Okay? You'll also notice the presence of the uterus, but that's really, you wouldn't call it retroperitoneal, but you basically would say it would be in more the pelvic area. Okay. Now, we're going to start in the mouth and move on downward. By examining the gross anatomy of the alimentary canal, we're going to start off with the oral cavity, go to pharynx and esophagus, and then move into the other organs. When we use the term alimentary tract, this is the muscular tube. It starts at the mouth and ends at the anus. The oral cavity is inferior to the nasal cavity and is separated from the nasal cavity by the palate. When I talk about the term bolus, that's a compact mass of food. You want to be able to, in diagrams and images, identify the following: the lips, also known as the labia, cheeks, tongue, gingiva, which is also referred to as the gums, the labial frenulum, the vestibule, the palate, the uvula, the lingual frenulum, the hard and soft palate. Also the fauces, the palatine tonsil, the root of the tongue, lingual tonsils, pharynx, which consists of nasopharynx, oropharynx, and laryngopharynx, and the esophagus. The opening in the diaphragm, which is the esophageal hiatus, this is where the esophagus passes through. The transport of materials through the esophagus is by a process known as peristalsis. That is a coordinated process that occurs. Now, at the end of the esophagus, you have the cardiac, otherwise known as the lower esophageal sphincter. Okay. Here we have the gingiva, the tongue, the fauces, which are the spaces in the back of the oral cavity, the uvula. We have the palatal arches, as you can see here and here. We have, of course, this is all that makes up the oral vestibule, the frenulum of the upper lip and the frenulum of the lower lip. There's also a frenulum of the tongue. That's the lingual frenulum. We have the palatine tonsil here, soft palate, of course, hard palate, and this is the ridges in the mucosa that are covering the hard palate. Okay? From a sagittal view, you can see where the regions of the pharynx are: nasopharynx, oral pharynx, laryngopharynx. This is the internal nares. So that's sort of the end of the nasal cavity. You can see the definition of the hard palate here, which is more bony, and then the soft palate here, which ends at the uvula. We have the esophagus here, which will basically flop down to prevent food as it's being swallowed from going into the larynx and into the trachea. The esophagus is right here. Now, examining the gross anatomy of the alimentary canal, we're going to start going into the stomach and the small intestine. The stomach is a J-shaped pouch in the abdominal pelvic cavity. We have mixing movements. These are muscular contractions to assist the chemical digestion, because you've got to basically mix up all these digestive materials, such as the hydrochloric acid and the gastric digestive enzymes pepsin, to provide sufficient chemical digestion of the food that is entered into the stomach. Eventually, we reduce the food to a paste-like material called chyme. Now, the stomach anatomy, different regions are referred to by particular terms, so we want to be familiarized with them. You have the fundic region, the cardiac region, otherwise known as the cardia, the body, which is the main portion. The pyloric region, which is also called the pylora, as it's a sphincter at the end, controls chyme delivery into the small intestine. And when I say the small intestine, the beginning of it is the duodenum. And we have, of course, the lesser curvature and the greater curvature of the stomach. Now, the muscle layers in the stomach are basically smooth muscle layers. The outermost is the longitudinal, the middle is circular, and the oblique is innermost. We have these tissue folds called rugae, which allow for expansion and contraction of the muscle walls. Of the stomach walls. And the small intestine consists of 3 sections, and you will have chemical digestion of complex nutrients occurring there. The end result is simpler molecules. You will have monosaccharides from disaccharides and polysaccharides. You will have amino acids from proteins. You will have fatty acids, basically from the triglycerides. And this will be absorbed into the blood or lymphatic circulation. Anything that's relatively longer, such as monoglycerides or fatty acids, that'll go by route of the lymphatic system. The amino acids and monosaccharides will be going via the route of the blood system. The small intestine anatomy, as I mentioned before, the first section, which is roughly about the first 18 inches, is called the duodenum. It forms a C-shaped structure. You have the jejunum, which is highly folded, superior portion of the intestinal folds. The ileum, which is the inferior portion of the intestinal folds. And then you have this unique structure you're going to see called the PLICAE circularis, which is the transverse folds along the inner walls. Causes a spiraling movement of the chyme through the intestine. And it's most prominent in the jejunum. This spiraling allows for further surface contact with the inner parts of the chyme so that nutrients can be absorbed. Here we have, of course, the fundus. This is the anterior surface, the cardia. The end here of the esophagus going into the stomach. The muscle layers of the stomach, the longitudinal, circular, and the oblique. This is the body area of the stomach, the lesser curvature, the greater curvature. We have the rugae, those folds that you will see on the inner surface. And then we come into the area known as the pyloric part, or the pyloric antrum, which consists of the pyloric canal, the pyloric orifice, which is the opening here, and that's controlled by the pyloric sphincter, which will, upon certain signals, neural or chemical, hormonal usually, will open or close, allowing the squirting out of some of the chyme into the beginning of the duodenum here. Now, when we show you this entire anatomy of the small intestine, you start off with the duodenum, then you go through a lot of the area of the jejunum, which is more the superior part, and the ileum is more the inferior part, which eventually ends at the ileocecal sphincter, where you're going to have the release of the material into and entering into the large intestine. Okay? The large intestine also goes by the term colon. Here you see the circular folds, the plicae circularis. So they would cause the material to go in sort of a circular spinning throughout the system. Now, when you examine the gross anatomy of the alimentary canal, we get to the large intestine. Now, the large intestine is different because the large intestine is not going to do as much absorption. There is a little bit, but a lot of it also is the removal of water and electrolytes from the fecal matter. The large intestine is larger in diameter but shorter in length than the small intestine. Feces are the undigested and unabsorbed material. So one-third of it will consist of bacteria. There will be, for example, undigested cellulose fibers and things like that. We remove water and electrolytes from the fecal matter, and then we get into the concept of defecation, which is the fecal elimination via the rectum and the anal canal. The large intestine site is very important for its microbiome. You see, what happens is there's a large microbiological ecology, an ecosystem in that large intestine. Some of these organisms break down the fiber. Some of these will actually synthesize certain B vitamins for us. Now, when we talk about the large intestine, the gross anatomy structures, we would talk about the cecum, the vermiform appendix, otherwise vermiform comes from the term worm-shaped. These are attached to the posterior medial wall of the cecum, the ascending colon, the right colic flexure, the transverse colon, and the left colic flexure. Also, we have the descending colon, the sigmoid colon, the rectum, the anal canal. Now, there are some structural specializations that I want to bring to your attention here in the large intestine. The tennae coeli, which is some very fine pathways of smooth muscle, but it's not completely circular. It's on a few areas only. The ileocecal junction and the ileocecal valve. This is where you're going to have the exchange of material going from the ileum into the cecum. Haustra are the series of sort of pouches or billowing out structures of the membrane of the large intestine. We also have on the exterior what they call fatty appendices. These are fat-filled tabs. And then, of course, we have the anal canal, and the anal canal consists of the internal anal sphincter. This is an involuntary smooth muscle. Now, the anal canal is relatively short, but what I want to bring up is the sphincters. The first sphincter is the involuntary smooth muscle, and that will get connected. We're not as conscious of it. The external one is a voluntary skeletal muscle. So the external anal sphincter there, that's the one that we all learn as little kids, how to hold on to it till we go to the bathroom, okay? Now, here you see the diagram. So here's the ileum, the ileocecal valve, okay? You have the vermiform appendix here, the cecum. As you move up, you have the ascending, you have the right colonic flexure. They also call it the hepatic because obviously the liver would be over around in this area. Then you have the transverse colon, the left colic, otherwise known as the splenic flexure, because the spleen would be over in this area. And then we have the descending colon, the sigmoid, think of sigmoid like an S, and then it empties into the rectum, and then we have the anus over here. Now, a couple of things to keep in mind. Notice that you're going to have the superior mesenteric vein and the superior mesenteric artery taking care of the blood needs. Now, I stand corrected there. There is a superior mesenteric artery right here that spreads out, that takes care of part of the large intestine. The other part is from an inferior mesenteric artery, okay? And you also have basically a complementary inferior mesenteric vein that will also take care of return of blood here. Notice that you have this tendonae coeli. These are a couple bands of smooth muscle here, so you don't have it going side to side or completely around the entire structure. You have these omental appendices, otherwise known as these fatty tabs here. You can see them here. You can see them here. And you can see how you have these sort of billowing or pouches, and this is the haustra, okay? When we get from the rectum, we're going to start going into the anal canal area, which has some anal columns. These are all sensitive to the stimuli because of the pressure as the feces are moving into here. The interesting thing that you'll also find is on the inside structure here, you have the internal anal sphincter. That is all smooth muscle. It's involuntary. The external anal sphincter, this is all skeletal muscle, and this will have, you will have conscious control of this and when to basically empty your bowels or do defecation. Here's the anus here. Now, when you examine the gross anatomy of the accessory digestive structures, this is going to include some of the glands in different parts of the body. So we're going to start off with the tongue and the salivary glands. The tongue forms a large part of the floor of the oral cavity. You have both intrinsic and extrinsic tongue muscles. But also, keep in mind, the tongue is used to kind of like mix together food in the beginning as you're grinding and chewing it up. But it also, you're going to have some mixing of the salivary gland secretions. These keep the oral cavity lining moist. There are serous glands and mucus glands. Serous glands produce a watery saliva. Mucus glands produce a mucus saliva. Now, let's examine the glands. The parotid gland secretes mostly a watery saliva and some amylase. If you remember amylase, that is a starch hydrolyzing enzyme. And we have a parotid duct, so this is part and parcel of the gland, which is an exocrine duct. It secretes from the gland via a duct into an outer lumen, in this case it would be your mouth. A sublingual gland secretes mostly a mucus saliva, and the submandibular glands secrete a mix of both watery and mucus saliva. And you can see them located here. So here we have the parotid duct, the parotid gland. We have the submandibular gland, and we have the submandibular duct, which comes up and joins the Wharton's duct, which is the opening of the submandibular duct, right here below the tongue. The sublingual is here, and it also contributes to this discharge, okay? So you have submandibular, sublingual, and parotid glands, okay? You will also see the diagram of the muscles. Since we've done muscles before, we don't need to do as much work on this, just to understand that there are both extrinsic and intrinsic that will help the tongue function in various movements during the process of how we start to process the food that we are consuming. When we examine the gross anatomy of the accessory digestive structures, we have to include, of course, the teeth. Teeth are embedded in sockets of the alveolar processes of the mandible and maxillary bones. We have two sets of teeth, deciduous, secondary. The deciduous, otherwise known as your baby teeth, the primary teeth, appear between 6 months and 2 years of age. The secondary are the adult teeth. They first start to appear about age 6 years and are completely in about between the ages of 17 and 21 years old. The 32 teeth consist of the central incisor, lateral incisor, cuspid, otherwise known as the canine, the first and second premolars, and the first, second, and third molar. Now, the third molar is also referred to as wisdom teeth because what happens there is some individuals' mouth are basically not big enough to handle that third molar. The erupting tooth can cause a lot of problems and sometimes has to be extracted. Now, we have to connect chewing function to it with the type of tooth. So the structure of the tooth is very, very important. We have the crown, the neck, the root, the dentin, the enamel, the cementum, the pulp cavity, the root canal, and hold off, root canal, we have a root, or basically a set of nerves that come out from the edge. And if we have dental caries, this is due to a developing hole because of bacterial acids that will wear away at the enamel and perhaps into the dentin. We have to remove the damaged material and place some sort of filler into it. Now, when do we have root canal therapy? That's going to be tricky. That's when you have to basically have where the pulp cavity has been damaged to the point where you have to basically remove it. You may have to also neutralize the function of the root. Now, when we remove teeth, first the dentist must cut the periodontal ligament. And a tool to do that is called a luxator. Then they remove the tooth via extraction forceps. So when you look at this, okay, here are the permanent teeth. So starting with the central incisors, lateral canine, first, second premolar, first, second molar. This is the tricky ones, the third molars, otherwise known as the wisdom teeth. Okay. Now, when we look at from a side view, you have, of course, the occlusal surface. This is the surface where the teeth meet other teeth, or basically do the grinding or crushing that's necessary, or cutting. You have the occlusal surface here, and it is covered, excuse me, not the occlusal surface, the enamel. The enamel covers over the tooth. Underneath is dentin. The enamel, interestingly enough, has a different embryological derivative than the tooth itself. And the tooth has a lot of these very fine tubes. You may have heard individuals when they talk about they are very sensitive to cold or heat or whatever when they drink something, and that's when they have these special toothpaste that basically what they do is they plug up these little tubes to prevent the exchange of hot or cold material to come into the pulp cavity and cause pain sensations. This is the root canal. This is the apical foramen, the opening that allows for the root canals as the blood vessels, arteries, and veins, as well as nerves coming through. Now, we have the cementum here, or cement here. We have the periodontal ligament here. This is what we also call the gymphosis joint, where the tooth and the bone do not move. So what do you have to do? You have to literally have a tool that comes in and cuts through this ligament and cuts through this side here so that you can be able to remove the tooth. Okay? And what you see here are luxators. These are severing the periodontal ligaments so they get in between the tooth. And then you have an upper right forceps. Now, the interesting thing is I took some dental training some time back, and they're actually, you can't use a force, you can't use like pliers. You see that on the cartoons, you can't do it. Because if you put the amount of pressure that a plier could be using on that tooth, it would crush the tooth, leading to a subsequent infection, failure to remove all the tooth components. It's a terrible mess. There are about 3 different types of forceps, depending on the angle, depending on the tooth that you're going to be extracting. Is it from the upper or lower mouth? Is it from the back or the front? Okay, that's just to help you to understand that principle there. Now, we've moved from the teeth and the mouth. We're going to start looking at the liver, the gallbladder, and the pancreas, because these are the accessory digestive structures. The liver is the largest internal organ in the body. Its primary function is the production of bile through emulsification of fat globules in the small intestine. Remember that if you have large fat globules, they will make it harder for lipases to break down the fats. So what bile acts is like a detergent. It removes large blobs of fats and reduces them to smaller fat globules so that lipase can more quickly break down the fat. When you look at the anatomy of the liver, it has a bare area, coronary ligament, the right and left lobes, the falciform ligament, the round ligament, which was also known as the ligament teres. Okay. The posterior inferior view, you would see the caudate lobe, the quadrate lobe, and the porta hepaticus. This is the junction of the 4 lobes. It is the entry and exit for hepatic artery, portal vein, lymphatics, nerves, and hepatic ducts. Now, when we talk about the gallbladder, keep in mind that the gallbladder is a temporary storage site for bile. It is located at the anterior inferior edge of the liver. The structures of the gallbladder include the left hepatic duct, the right hepatic duct, the common hepatic duct, the cystic duct, and the common bile duct. Now, what I want you to understand is this. When bile goes in and is stored there, you've got a mixture of cholesterol and ions and water and bile pigments, etc. Now, what is going on? As the bile stays there for a period of time, you slowly begin extracting out water and some of the ions. And as a result, it can lead to the precipitation of the solubilized cholesterol and the bile salts. This can lead to the formation of, gallstones. And you can live without your gallbladder. Many treatments today to deal with gallstones are basically remove the gallbladder with the stones. And a lot of this is done with smaller tools. And so the patient can recover much more quickly. But the only difference now is that they have to be told you've got to be on a much more reduced lipid diet. You can still have fats, but you have to be in a much more reduced amount because you're not going to have any spare extra bile to solubilize those fats. So if you have a whopping amount of it, let's say, hot fudge sundae with lots and lots of whipped cream, you might end up having steatorrhea, which is lipids in the feces because they did not get broken down in by the interaction of the bile and the lipase in the small intestinal system. Moving forward, we're going to talk about for a brief time the pancreas, which secretes pancreatic juice, which is a mix of digestive enzymes and buffers. Delivery to the small intestine of this juice for digestion of nutrients as well as the neutralization of chyme acidity. The thing you have to keep in mind here is that when chyme leaves the stomach, it's been saturated and mixed with hydrochloric acid. Hydrochloric acid pH. In the stomach, it will be about a pH of 2. Okay, so if you don't neutralize that strong acidity as this material is going into the duodenum, it can start to cause damage to the lining of the duodenum. So we release this juice, as we would call it, the pancreatic juice, which the buffers are predominantly bicarbonate ions. You will also have digestive enzymes, and these bicarbonate ions would, of course, neutralize a lot of the acidity. The structure of the pancreas, now remember, this is a dual exocrine endocrine organ. Here we're talking about the exocrine capabilities now. The structure is head, body, tail, pancreatic duct, duodenal ampulla. Here you can see, as you're looking from a horizontal section at the level of T11, you have the liver, okay, the different lobes of the liver. You will also see nearby, of course, the stomach and the spleen. And you can see this from an actual cadaver image. And you see some of the larger holes there, not artifacts, but are really larger blood vessels that are in that liver. Now, if you look at the image here, this gives you the idea. If you think about the falciform ligament, sort of like a dividing area between the right lobe and the left lobe, and part of this is also the round ligament here. You can see just peeking out here the gallbladder. This is the bare area here. And if you go to the visceral surface, not the anterior surface, but underneath, you can see where the location of the gallbladder is, the proper portal vein, you have the portal artery, the porta hepaticus, you've got some of the bile duct here. You can see the bare area here and a coronary ligament that kind of cuts through this. You have left lobe, the caudate lobe here, the right lobe, and then you have the quadrate lobe here. Why is this important? Well, for one thing, you can see how large this organ is and how close it is to such main structures as the inferior vena cava. But the other thing is this, every once in a while you'll hear someone saying, I'm going to donate a part of my liver to someone else so they may live. This is a much more recent technological advance where you can actually take apart certain parts of the liver and they will be functional in someone else as long as all of the histocompatibility antigens and genes and everything else are worked out. And the donor will have their amount of liver that still maintains their function for the rest of their life. Also, even if there is some liver damage, some liver has a greater capacity to recover as an organ. Here is up close the gallbladder. You'll notice the fundus, the body, the neck, the cystic duct. Okay? And then, of course, you've got the common hepatic duct, the right and left hepatic ducts. They are going to follow down. You have the bile duct down here, as you can see, following it down into the diagram until you get to this main site, which they would call here, well, it was also by the name of sphincter of Oddi, O-D-D-I, but I believe they call it the duodenal sphincter. But we'll get into that in a minute. You'll notice also as you pass through this, because you are going through some of the lesser omentum, that you have the hepatic portal vein, the hepatic artery proper, right here. And of course, tucked into this fold here, from stomach to duodenum, is the pancreas itself. Now, they call this the duodenal papillae. This is really the junction of the pancreatic duct with the bile duct. Now, there is an accessory pancreatic duct that allows a discharge here if this one was blocked. But if you notice, there is this ductwork that goes through the body of the pancreas, and it collects the secretions of the bicarbonate ions and the inactive enzymes. They will be activated when they get into the duodenal lumen right here. This is the tail of the pancreas, and all of these are lobules consisting of acini, and these are the cells that are going to make the various secretions, the enzymes, etc. Okay? Now, when you examine the microscopic structure of the alimentary canal, let's take a look first at the esophagus and stomach. The esophagus, as we've talked about before, is basically transporting food from the pharynx to the stomach. You have from the inner lining, where you would have, of course, the lumen, you have mucosa, and that you're going to have basically stratified squamous epithelium. It has to be stratified because you're going to have erodive effects as the bolus of food is pushed along through the esophagus. You will have underneath it lamina propria. Underneath that, the muscular layer of smooth muscle, which is called the muscularis mucosa. Underneath that, of course, you're going to have a submucosa of esophageal glands, connective tissue, blood vessels, and nerves, and then you have the muscularis externa. Now, you will also have an adventitia, which is the outermost covering for the esophagus. With the stomach, this is going to contain numerous gastric pits. They're deep in the mucosa membrane and lead to the gastric glands. The mucosa is going to consist of gastric pits, and they're lined with simple columnar epithelial cells. The gastric glands produce gastric juice, which is a mix of hydrochloric acid, mucus, and digestive enzymes. Underneath that, you will have, of course, the lamina propria, which is connective tissue. You will also have layers of the muscularis mucosa, the submucosa, the muscularis externa, the serotia, and don't forget that on the inner lining, you're going to have these somewhat stomach folds, which are called the rugae. And you can see here, this is our esophagus first. You see this mucosa here, which is non-keratinized stratified squamous epithelia. So the upper level cells will be stripped off as boluses of food move down, but underlying cells would replace them. You have the lamina propria, and then you have the muscularis mucosae. Okay? The muscular layer is beyond that, And this is all smooth muscle, and then you have an outer lining of adventitia. When you go into the histology of the stomach, you're going to notice that you have simple columnar epithelium. It secretes an alkaline protective mucus right here to protect it. Okay? You have a gastric pit, and then into it you're going to have these glands, the glandular cells that are going to produce various substances. Underneath it, the lamina propria, the muscularis mucosa, the submucosa, and then the muscularis externa. All of this are the different types, the oblique muscle, circular muscle, longitudinal muscle, and then you have the serosa, which is really the consisting of the visceral peritoneum. Notice the blood vessels do rise up into some of these column-like structures of tissue, okay? And if you notice in the stomach, here's the surface mucous epithelium. You have these pits, the lamina propria, the glands, okay? It is really important to keep in mind that even though you're producing a tremendous amount of acid, you have to have protection so that the acid doesn't damage the lining of the stomach. Moving to the microscopic structure of the alimentary canal in the small intestine, we find that it is highly folded to enhance surface area for nutrient absorption. That's the key point I want to walk away with. The plicae circularis are folds of mucosa and submucosa. Then on top of those, they have further extensions of the surface area by the intestinal villi, which are finger-like projections along the surface of the mucosa. And we have, going down deeper, deep pockets, these intestinal glands. They will secrete mucus and numerous digestive enzymes. The muscularis externa contains inner, circular, and an outer layer, which is considered longitudinal, of smooth muscle fibers. They allow for peristalsis as well as segmentation, which allows for a mixing of the materials. Peristalsis, of course, is the synchronized means to push materials down the length of the intestine. When you get to the villi microanatomy, you're going to find a bunch of different types of cells that are very important. You have the absorptive cells, that's the main cell type. You have goblet cells, obviously, they're going to produce mucus. You have the enteroendocrine cells, which are located in the epithelium. They are going to release certain hormones that will trigger certain activities and coordinate these activities between stomach, liver, pancreas, as well as the intestine. Okay? Of course, in each of the villi, you're going to find blood capillaries. Also, you will find a lactea, which is a lymphatic capillary. Remember, the lymphatic system is going to transport also via the lymph some of the more trickier lipids. And I say tricky, it doesn't mean that. It's just they would not be able to be immediately transported into the blood capillaries. What would travel in the blood capillaries would be, again, amino acids and monosaccharides. Now you have an edging on the villi, and this is composed of the microvilli to enhance absorptive area, and this is called the brush border. Now there are sectional differences between the different parts of the small intestine. In the duodenal glands, they secrete an alkaline mucus. In the jejunum, the intestinal villi are most prominent, so you have a lot more of the absorption occurring there. And that only makes sense because after the duodenum, you started really breaking down a lot of the substances that you're going to eventually want to absorb. In the ileum, you notice a profound number of aggregated lymphoid nodules. We also call them Peyer's patches, which are involved with monitoring if there are any pathogens and other toxins, etc. So if you look at this, you go from one level to the next. First, you have the plicae circularis, and on the edges of the circular folds, you have villi. On the edges of the villi, you're going to have eventually the microvilli here, the brush border. Inside of the villi, you're going to have, of course, the lactea, which is the lymphatic capillary. You're going to have also capillaries, which are going to absorb some of the nutrients and transport them. In the mucosa, you're going to see this structure. You're also going to see these intestinal glands are called intestinal crypts, which may also produce some mucus. as well as some cells that are going to produce certain hormones. Now you have the muscularis mucosa. You also don't forget that lymphoid nodule there, which monitors the immune structures and if there's any problems. Underneath the submucosa, and the submucosa is going to have arteries and veins there. Then you get to your muscular layer. And there you're going to have both circular layer of smooth muscle as well as a longitudinal one. And you're going to have the coordination of some of this activity by the myenteric plexus. And this is this nerve pathways that will coordinate certain contractions and the timing of them, etc. After you get through the longitudinal layer, you are basically at the serosa, which is the outer layers. Okay? Now, moving on. Here is a light microscopy image at 250 magnification. You can see the lacteal here. This is the tip of the villus. So you can see the capillaries that are right in through here that are going to absorb the monosaccharides and the amino acids. The lacteal here will pick up the monoglycerides as well as the fatty acids. Goblet cells are adorning the top part here so that they can basically secrete mucus. Okay? If you move to this image, you see all of this connected. Here is a villi, and on the tip of it, you can see the little fuzzy little hairs. That would be the microvilli. Here's your goblet cells, your capillary network, and it surrounds around that lacteal. Okay? Moving on. Here you see some of the structural differences as you go from the duodenal. Notice that you have some shorter villi. The jejunum, which you have much longer patches of villi here. The ileum, you have a reduced amount of villi, but you do have these pair patches, and that's what the PP is for. When we examine the microscopic structure of the alimentary canal at the large intestine, you're going to notice some distinct differences. The inner wall will lack intestinal villi. The mucosa, it has simple columnar epithelium. It has some invaginations, intestinal glands, lamina propria, muscular mucosa, and mucus cells. When we get to the submucosa, we go from there to the muscularis externa. We have a serosa that includes the tene colae. This is the longitudinal smooth muscle layer. Okay, so you don't have these large layers all the way throughout, all around the entire circumference of the large intestine. Here are the crypts of Lieberkühn. Here's the lumen. The crypts are where you would have some secretions occurring. You have, of course, the mucosa here, submuscularis mucosa here, submucosa here, the circular muscle here, and you have some of this, the tene colae here. These are just sort of straps or strips of longitudinal muscle. When we start examining some of the accessory digestive organs, first, of course, we're going to deal with the salivary glands. There are differences between the serosal cells and the mucosa cells. Remember, serosa is going to make sort of a watery type of saliva. The mucosa is going to be more a mucousy type of it, so its viscosity is greater. We have, of course, the salivary gland duct, and the gland cells are called acini. Here we have the duct, and you can see how we have sort of like almost the simple, rounded epithelial cells. Here, you've got the mucus cells, and they're just producing large amounts of mucin that will be pumped into the ducts. The serosa cells are much different. They stain darker, and you can see somewhat this rounded tubule structures here and here. Okay, this is found in the submandibular gland. When we look at the accessory organs of the liver and the pancreas, we have to examine the liver lobule and identify the various structures as follows. The central vein, the portal areas, which is known as the portal triad. Okay, in the portal triad, you will have a hepatic portal vein, a hepatic artery, and you will also have a bile duct. You will also notice lots and lots of hepatocytes, as well as the liver sinusoids, the bile caniculi, which is the transport of bile through the liver, and Kupffer cells. These are phagocytotic cells embedded in the walls of the sinusoids. We'll also notice that when we go through the pancreas, it's very straightforward. We're making pancreatic juice, which contains digestive enzymes and bicarbonate ions. Okay. The pancreatic acini are basically clusters of cuboidal cells arranged around a central lumen. Now, that's very different from the pancreatic islets. Remember the islets of Langerhans? The islets are going to stain lighter, and they are purely endocrine cells. The acini stain darker, and they are exocrine cells. So here's a diagram first of the liver. Here's your portal triad, okay? So you're going to have interlobular bile duct, interlobular vein, interlobular artery. That is the portal triad. They all seem to be almost like in a hexagonal arrangement, portal triad here, 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 and here, and you have this central vein-like structure here, but also you have lots of cells here that are going to process chemicals, and when you get up close, you will see several other structures. From the central vein, you'll also notice that there are little tubular structures in between the cells. These are the bile caniculi, okay? And what's going to happen is they pick up the bile salts that are made. You will also have these structures passing through the venous collection ports, and they are called stellar macrophages, but their other name really is Kupffer cells. They are fixed macrophage structures that will remove certain toxins or pathogens, okay? And you can see how these hepatocytes are basically, this would be one row right here. This would be one hexagonal section of them. You can see this using the slides. Here's your central vein. Here is your portal area with your bile duct, your hepatic artery, your portal vein, okay? And these are all hepatocytes throughout here. And you have your lobule boundaries right here, here, here. With the pancreas, you will have a pancreatic duct. You will have these round clusters, the acini, and they will be producing and secreting the bicarbonate ion solution as well as enzymes. Notice that the difference that they've kind of lightened this up, and this is going to be your endocrine cells, your pancreatic islet. And don't sweat it. I'm not asking you to identify which one's an alpha, which one's a beta, which one's a delta, and which one is an F cell. That's something that would be much more difficult to do by just looking at cells. You have to have usually immunohistocytochemistry present, okay? Just understand that for the pancreas, you have acinar cells, okay? Here you would have your acini. Here is your acini here in this image, and you notice that this is lighter staining here. These guys are darker staining. This is your islet of Langerhans with the cells that produce the various types of endocrine secretions we would call hormones, okay? All right, we're pretty much done here. So what do you have to do? You would want to take the time to review the gross anatomy of the digestive system and any microanatomy images of the digestive system, as well as accessory digestive organs. Now, key points here, you're going to be able to do the review sheets. These are the worksheets that I have scanned in. Fill in the different sections, upload it again into the Blackboard site. Never send me a copy because I could have somebody sending me something inadvertently that has a virus or a Trojan, and if my system crashes, then it causes all sorts of chaos, and I don't like that. Rather, upload the work so that we have evidence that the work is done and I can effectively and quickly grade it, okay? And get this in by Friday at 5 p.m. and everything will be fantastic. What will help you will be the lab videos. as well as reviewing that section of the lab exercise sheet, or the lab exercise book, I should say. Well, have a nice week, and we'll see you soon.